Good afternoon, students. This session for second year Biotechnology students, Semester 4, paper Plant Biotechnology. Today topic is sterilization in plant tissue culture media. So in this slide, you can view left side corner is the hot air oven, then down in the bottom we can see the plant material that is called surface sterilization and autoclave. So right side of the corner plant tissue material is present. If you see, one part of the plant material is very good without any dust particles, and other part right side is some dust particles are present. So if you are going to plant tissue culture, first of all we are sterilize the leaf material. Here without sterilize the plant tissue or plant explant can inserted into the appropriate media, they can destroy. That's why here we are using the sterilization techniques. So first of all, what is meant by sterilization? The simplest definition is sterilization is defined as the process where all the living microorganisms, including bacterial spores, are killed. The meaning of sterilization: microorganisms are removed or killed by the sterilization process. Sterilization can be achieved by physical and chemical methods. Chemical methods used as sterilizing agents are called chemisterilants. Here, the sterilization methods are the steam sterilization by using autoclave. Here 121 degrees centigrade at 15 psi for 20 to 40 minutes. Nutrient agar. On that nutrient agar media having peptone, yeast extract, sodium chloride and agarose, distilled water. If you are preparing the media, then the time any microorganisms are intact on that media, prior to going to plant tissue culture steps, the media can sterilize by using autoclave method. Autoclave ద్వారా స్టెరిలైజేషన్ చేస్తామండి. కల్చర్ వెజల్స్. These culture vessels are nothing but conical flasks or Erlian Mayer flasks. Then glassware and plasticware. Glasswares are nothing but here measuring cylinders, petri plates, test tubes, so pipettes. These are the glasswares. Then plasticware. So plasticware also used. Dry heat method, hot air oven 160 to 180 degrees centigrade for 3 hours. Instruments like scalpels, forceps, needles. Then glassware, pipettes, tips and other plasticwares. Flame sterilization instruments. Under the Bunsen burner or flame, scalpel, forceps, needle can be sterilized. Mouth. Then filter sterilization. Here membrane filter, it is made up of cellulose nitrate or cellulose acetate 0.45 to 0.22 micrometer porosity. Thermolabile substances like growth factor, amino acids, vitamins and enzymes. Alcohol sterilization. So in alcohol sterilization, here plant tissue culture technician, when enter into the lab, before going to the lab, first of all he can sterilized with the help of sanitizers or sterilant. Then only the samples can survive. Without sterilant, without sanitize, the technician entered into the plant tissue culture lab. They maybe the cultures are decomposed, or the cultures are contaminated. That's why before entering to the plant tissue culture lab, first prior to sterilize itself. Then alcohol sterilization, worker hands, laminar airflow cabinet, surface sterilization like sodium hydrochloride, hydrogen peroxide, mercury chloride. These can be used as a excellent surface sterilization by plant tissues. Then here, what are the types of sterilization techniques in plant tissue culture? So here mainly 5 types of sterilization techniques. First one is the sterilization by heat method. Second, sterilization by filtration. Third, air sterilization. Fourth, tunnelization. And fifth, surface sterilization. So here, the first one is sterilization by heat method. In this sterilization by heat method done by hot air oven, dry heat and moist heat are used to sterilize the equipment. Here, dry heat and moist heat both are used in the hot air oven. Then autoclaves, they work on the principle of moist heat. So LBs, they are represented in LBs, and they can sterilize glass apparatus, solid or liquid media, distilled water, normal saline, discarded cultures, and contaminated media. Presence of autoclave. Then, whereas hot air oven work on the principle of dry heat method. Dry heat method here used as a glasswares. Then these glasswares present in the slide, they are conical flasks, Aurelian and Mayer flasks, pipettes, test tubes, glass slides, bakers, measuring cylinders, conical flasks, then measuring cylinders, and bakers. Okay, then they can sterilize glasswares like petri dishes, conical flasks, pipettes, and test tubes. Here, some chemicals like used as a powders like starch, zinc oxide, sulfidizine. That materials contain some oils. Metal equipments like scalpels, scissors, blades. These all things done by sterilization with heat. Here, in this heat, moist heat and dry heat. Dry heat in the presence of dry heat is presence of hot air oven, moist heat present in the autoclave. Then going to the second slide, sterilization by filtration. Here sterilization, filtration in cellulose membrane filters are used. Here a liquid solution is forced through a membranes to sterilize it for microorganisms. Here test sample can transferred via filtrate. Here some microorganisms are trapped. Then through filter membrane, finally we get the filtrate molecules. So here the size of the pore in the filter decides what size of microorganism will be filtered out. So the required amount what we need, then the porosity containing filters are used. Then it is used to sterilize growth substances like thermolyable zeatin, then gibberellinic acid, abscisic acid, urea, and certain vitamins are used as a sterilized growth substances. Then going to the third slide, then third type of sterilization is called air sterilization. In this air sterilization done by laminar airflow. So in this laminar airflow is good instrument to handle or to transfer the media presence of laminar airflow. Here first UV light is present, then UV light, when the UV light is on the microorganisms, some microorganisms may kill, then alcohol sterilization, wiping the alcohol, then only visible light. So here high efficiency particulate air filters are used. In these particulate filters are blower that blows the air through the filters to prevent all sorts of the microorganism larger than 0.3 microns with 99% of efficiency from the air. So here the particles like bacteria, viruses, some fungal particles are uh, uh, coming and out, in and out by using the filter through air. So these are called airborne microorganisms. They can cause airborne diseases. Here air sterilization with the help of blower. In these blowers can receiving the air particles. The next one, thermalization. This thermalization is called as fractional sterilization. It is used to kill heat resistant endospore. It is used to heat resistant endospores. So this was first introduced by John Tyndall. That's why his name mentioned, his name coined as the thermalization, like as Patcher effect, introducing Louis Patcher. That's why Patcher effect. Like in this technique, the medium is heated in a water bath for 1 hour for 3 consecutive days and then kept at room temperature after 2 incidents for successive boiling, like pickling of the pickling and some of the food preservatives, like using thermalization process. It is called fractional sterilization. And the next one is the surface sterilization. In this surface sterilization process, plant tissue culture method, this technique is used to sterilize the surface of the explant. So here meristem or leaf particles. So you can use in your tissue culture process. So then cutting the tissue, then it was soaked in particular solutions like mercury chloride and sodium hypochlorite or hydrogen peroxide. By using these chemicals, any unwanted particles bound to the plant material, they can removal by using the rinsing of the material, the washing of the material with the help of appropriate solutions. So here the surface means the plant containing the upper both dorsal and ventral side, the plant particles having the plant particles can attach any dust particles like organisms are attached or present in the leaf. They can eliminate or they can removal by using the solutions like this. Then common explant sterilization. So by using mercury chloride, it is very rarely used in labs because it is high toxicity to plants as well as humans. So if you are using it, follow extensive care. After exposing the explant to mercury chloride, rinse them many times using sterile water to remove all traces of mineral from the explant. Second, sodium hydrochloride. In this, sodium hypochlorite is commonly known as bleach. It is most frequent choice for surface sterilization of explant. Commercial laundry bleach is 5.25% sodium hypochlorite. When used in the tissue culture, bleach diluted 10 to 20%, that results in 0.5 to 1%. So here the duration of the time and it can be avoided some materials. Third one, calcium hypochlorite. It is commercially available in the form of powder, and before using its sterilization, it must be dissolved in water. After dissolving in the water, filter solution. The final concentration of the solution used in the labs around 3.25%. Hydrogen peroxide, it's a rarely used chemical for surface sterilization. The favorable concentration of using hydrogen peroxide is 30%. Take all precautions if you are using the chemical for the surface sterilization of your excipient. And finally, used ethanol. It is also called ethyl alcohol, C2H5OH. The 70% ethanol is extensively used as a sterilizing agent in the tissue culture labs. But it's also extremely phytotoxic. That's why excipients are only kept for a few seconds in the chemical. The longer exposure to ethanol damages the tender excipient. So here these are the steps we are using the sterilization process. So sterilization steps are the 5 types: by heating method, filtration and air sterilization, analyzation, surface sterilization. These all are used for the ప్లాంట్ టిష్యూ కల్చర్ ల్యాబ్స్. థ్యాంక్ యూ స్టూడెంట్స్. దీనికి సంబంధించిన నోట్స్ కూడా నేను మీకు వాట్సాప్ గ్రూప్లో అప్లోడ్ చేస్తాను. క్లాసెస్ అనేవి రెగ్యులర్గా వినండి. నెక్స్ట్ మీకు ఎగ్జామ్ మీకు నోట్స్ అనేది కూడా నేను అప్డేట్ చేస్తాను స్టూడెంట్స్. థ్యాంక్ యూ. మీకు ఏమైనా డౌట్స్ ఉంటే కనుక యూ కెన్ కాంటాక్ట్ మీ. థ్యాంక్ యూ స్టూడెంట్స్.